In this video, I will explain the general idea behind single sample gene set enrichment analysis, and I will give a toy example to demonstrate how the enrichment score is calculated. This method is an expansion of the regular gene set enrichment analysis, which is used to understand how significantly a gene set or a signature changes between conditions. The conditions can be naturally occurring phenotypes or they can be treatments. And the signatures can be genes in a pathway or genes with common ontology terms or genes targeted by the same transcription factor. Really, any of the many gene signatures that can be downloaded from the molecular signatures database. And for the single sample version of GSEA, what the score represents is how consistently the signature genes rank highly or lowly in a given sample. The original paper describes the method as the integration of the difference between the cumulative distribution functions of the genes in the signature and the remaining genes. And we can intuitively think of this as the area between the two curves. One thing to note is the area is not calculated as the absolute difference. So if the direction of the difference changes, as in the case in this figure, these two areas cancel each other, and we get a score that is close to 0. So let's say we have measured 10 genes in a sample and ranked them, and the signature we want to score corresponds to genes with ranks 3, 7, and 10. I'll first talk about the second part of the enrichment score function from the original paper. This part involves genes outside of the signature, the yellow genes here. And for each gene, what we do is we count the number of genes its expression is higher than or equal to. So for the first gene, it's all of them, 10. For the second gene, it's 9. For the fourth gene, it's 7. The fourth gene is higher than or equal to 7 other genes, etc. And we sum these values and divide the whole thing by the number of terms. And this gives us the second part of the equation. The first part is similar, but we introduce a weight for each term. The weights are the ranks of the genes corresponding to the term raised to an exponent alpha, and also the denominator is no longer the number of terms, but the sum of the weights. What the weights accomplishes is, because data from a single sample is very noisy, it represents convoluted signals from a lot of different biological states. If there are extreme gene expression values that support the activation or a deactivation of a signature, the extreme value will have a more prominent effect on the score. For example, here we have the most lowly expressed gene of the sample, number 10, and it makes the denominator larger, having the weight here. So the score overall gets smaller. As a comparison, if we go back to the case where there are no weights, the first part of the equation would equal to 4.3, but with the weights, it becomes smaller, 3.36. And the exponent alpha is a parameter that can be changed. It determines how strong the effects of the weights will be on the score. The default value that is recommended, in fact, strongly recommended on the gene pattern website, is 0.75. So the final score is calculated by subtracting these two values that we have found. And for this particular gene signature, it's a negative score. If our gene set consisted of the 3 highest ranked genes in the sample, the score would be positive. And if it consisted of the 3 lowest ranked genes, the score would be, again, negative, but with a larger amplitude.